Okay, right now we're going to talk about the three types of protein-mediated transport that we discussed in the class this week, being diffusion through channels, carrier-mediated diffusion, and active transport. To start, just to kind of give an idea right here. So here's like a giant container I have, and I'm separating that container with a little thin semi-permeable membrane. We're going to call this side A, we're going to call this side B. So on one side, I add a lot of whatever this is. Just a few kind of like 4 different particles, right? Because everything has kinetic energy, everything's moving, slightly. And everything's going to want to reach a point of equilibrium where everything's pretty much the same on each side. So with time, this semi-permeable membrane that allows the movement of these 4 particles will eventually allow equal concentrations of both of the particles to be on both sides, from the A to the B. Okay, so now we're going to talk about diffusion through channels. Diffusion through channels is one way that we're able to move these particles, or we can call them solutes, either in or outside or leaving the cell. So here's a little cell right here. I'm going to draw a few of the different types of channels that we see within our cell membranes. So we have right here just a channel protein. So this is just a normal channel. Let's just say it's just a potassium channel, right? With these channels, they're very important for allowing specific solutes to move down their concentration gradient from high to low. So for example, for potassium right here, there's a ton of potassium within the cell. And there's not very much outside, so I'm going to draw a little tiny potassium. It's going to want to reach a point of equilibrium, which is where there's equal concentrations of potassium inside and outside of the cell. So this channel protein allows movement of this potassium to go outside the cell. With these channel proteins, they can be for specific ions. So in this case, it was with potassium. Other ones only allow sodium, which is Na+. Others will only allow chloride, and so on. There are other types of these protein channels as well. There are ones that have little gates on them. So you have your little one right here, and some of them just have a gate to prevent things from going from their high concentration to their low concentration. There are ones that are voltage gated, so changes in voltages will allow that gate to open. There's also, I'm just gonna draw them like this actually. There's specific signal molecules or gates where you need like a specific, I'm just gonna draw like something like that, where something needs to bind to this gate for it to open. And then there's other ones that are called mechanically gated channels, where there's like a stimulus such as like stretching, compression on the membrane, whatever it is. And I'm just gonna go like, I'm gonna draw something like that too. Okay. But these types of channels, so the diffusion through channels, they are a passive process. What they use is the concentration gradients from high to low to move the solutes. And when I say passive process, it means that ATP is not required. Kinetic energy alone will allow the particles or the solutes to move from a concentration of high to low to reach equilibrium. Things that can affect the rate pretty much of these channels, this diffusion through channels, could be the concentration gradient of the solute. So they say there's just like so much over on this side and so little over here. It's gonna be way more enticed to move over to that side. as if they were already, instead of if they were just already pretty close in concentrations. And then the number of channels. So if I have more channels that could allow, for example, potassium to leave the cell, it's going to happen a lot faster. Sweet. I'm just going to move down. Okay. Now we're going to talk about carrier mediated diffusion. Sorry, I'm trying to write this quick. Okay. So with carrier-mediated diffusion, carrier proteins bind to specific solutes on one side of the membrane, and the binding causes a change in the shape of the protein, which moves the whole protein, carrying that solute to the other side of the membrane. So it's kind of pretty much the same thing. So you have your cell right here, and I'm trying to move in, I don't know, let's just say sodium, for example. I have a little protein right here, and it's got a little spot for sodium to bind onto. Sodium won't be able to get through this membrane because it's a phospholipid bilayer. It doesn't let many things really get through unless through it like a channel or through proteins getting onto the other side. So this protein will bind to the sodium and move and cross over with the sodium to the cell. Or just whatever solute really you're talking about. And it could be moving from the cell to the outside of the cell, or from the outside of the cell to the inside of the cell. So I'll just say solute right here. Things that can affect pretty much the rate of these as well is the number of carrier proteins. So it reaches a point where, one second. Okay, so the rate of your diffusion here in carrier-mediated diffusion, or called facilitated diffusion, it'll go up as a concentration. So like as I add more of my solute, it's still going to be going through the high to low diffusion there. But since we're depending on these carrier proteins to allow the solute to cross that membrane, it's going to really depend on how many carrier proteins we have. So it's going to go up at a constant rate, but once we're using up all those carrier proteins, it's like, crap, we don't have any more to give. Like, we used our whole team, we can only go so fast. So it kind of reaches a point where it just levels off, where it can go only so fast. Even though we have like say we're in an assembly line, handing everything, carrying these things, like all this food, to another side of the whatever building you're in. And then even though there's a ton of food, we keep adding a lot more food. Trucks are coming with food, but we only have like 10 workers. We can only go as fast as the 10 workers can move, really. So that's how I kind of think of it. Perfect. And then, so yeah, with carrier-mediated diffusion, it still doesn't use energy. It still relies on a concentration gradient. So no ATP here is needed, really. Okay, and one thing that can also affect the rate is competition. So if there's a lot of different, if there's several different solutes, so say solute A and solute B, that can bind to the site right here, they're going to compete for that. So let's just say that solute A is a little bit better at binding at this spot. Its rate is going to be faster than solute B because it's just not as good as solute A as binding to this carrier protein. So if they're both competing, solute A is going to win. It's going to be a lot harder for solute B to go faster. Okay. Now we're going to talk about active transport. So there are two types of active transport. First, we're going to talk about primary active transport. Okay. The main thing with primary active transport is that it does require energy because it is moving the solutes against their concentration gradient. I'm just going to do this for concentration. their concentration gradient. One example of this is a really important one actually. It's our sodium-potassium pump. So I'm going to draw it right here. So inside the cell, we have a lot of potassium. And then on the outside of the cell, there's generally a lot more sodium. Perfect. So we have our pump right here. Let's just draw it right here. So with our sodium-potassium pump, it puts out 3 sodium ions and then brings in 2 potassium ions. And so for these, there's already a lot of potassium in here. Normally, since there's a lot more potassium inside the cell than there is outside the cell, you would expect that potassium would want to leave, right? But with the full help of ATP, we get help from the ATP to be able to push against this concentration gradient and push the potassium into the cell. And it's the same thing with sodium. Normally you would want sodium to go in with that concentration gradient, but we're pumping against that, and that ATP is going to provide that energy for that. Awesome. Yeah, and it'd be the same thing for specificity. So there's certain types, for all these, there's certain types of proteins that will bind to different things. So there's, or even the channels, only certain things can go through those, such as like up here. Like you have like a potassium leak channel. Only potassium can go through that. You have a sodium one as well. Only sodium can go through that. So things are very specific and in order. There's also something called a secondary active transport. Active transport. So this is also going against a concentration gradient. That's a long word. Okay, I'm just gonna stop right there. So against a concentration gradient. It's kind of like you're trying to push up a hill, right? It's a lot easier if you're just trying to run down the hill. You can roll down and it takes no energy. But if you're trying to go up the hill, it's just like, oh my gosh, that's like so tiring. It takes a lot of energy. It's the same thing here. You need some source of energy. The thing with secondary active transport that separates it from primary is that it's not directly using ATP as an energy source. What it is using is energy stored in the concentration gradients to move the desired solute that we're trying to move. So before we were talking about the sodium potassium pump right here. And what this is doing is it's creating that even bigger concentration gradient. It's creating even more sodium on the outside than the inside of the cell, and the same thing with potassium on the inside of the cell than there is on the outside. So this is creating even more of a drive for those sodium to want to go inside the cell, right? So, which was created by the use of ATP originally. So since sodium is in higher concentration outside the cell due to the primary active transport, the sodium gradient is typically used to power the secondary active transport. Carrier proteins, so they'll also still use carrier proteins right here. Let's just say, bloop, 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 right there. That's at the attachment site. So they'll have an attachment site for both sodium right here, and then for whatever other solute we want to use. Cool. So what the secondary active transport does is that this will bring the sodium into the cell, allow it to use that concentration gradient where it's like, there's so many of us out here, we want to get in there. So it'll just make its way inside the cell, and it'll bring the solute with it. This would be called a symport. So if something's going in at the same time with another solute, like the sodium, it'll be a symport. But this also can do the same, where sodium will use that concentration to go into the cell, and it'll drive out whatever solute we want, a specific solute. And that would be an antiporter or an antiport. But yeah, the main thing with this one is that ATP is needed to generate the sodium concentration gradient. but it's not directly involved in moving the desired solute across the membrane. And that's pretty much it with this part 2 with protein-mediated transport.